ये तो सेकंड पार्ट है, वो पीछे वाला फर्स्ट पार्ट, ये तो सेकंड पार्ट है। हाँ, ये फर्स्ट पार्ट है। फिर सेकंड पार्ट यहीं से शुरू हो जाएगा, उसके लिए फिर अलग फाइल खोलना पड़ेगा।
नहीं सर, सेकंड फाइल कहाँ पर है सर?
नहीं, यही तो है, इसको आपने दो पार्ट में लिया ना। नहीं, एक ही पार्ट में, इसको लास्ट तक देखिए तो। लास्ट नहीं, ये नहीं, आप मिनिमाइज करके लास्ट तक देखिए कहाँ तक जाता है, स्पाइनल कॉर्ड आता है कि नहीं। मैं सोचा आपने 1 to 30 लिया।
स्पाइनल कॉर्ड
हाँ, स्पाइनल कॉर्ड कहाँ तक आया, ये पूरा आया।
पूरा आ गया।
पूरा आ गया, ठीक ठीक।
शुरू कर दूँ?
हाँ, शुरू कर दीजिए। हाँ, अभी क्या स्ट्रीम कर रहा है?
हाँ, एक मिनट सर, मैं देख
हाँ, मतलब लोगों के पास वो बिस्लरी का बोतल है क्या पानी पीने का? हाँ, उसको। हाँ, स्ट्रीम हो रहा है, शुरू कर दूँ मैं? अच्छा, ठीक। Okay, welcome students to today's lecture, probably the first lecture on the nervous system. So the topic for today's lecture is the external features of the brain and spinal cord. So plus I will also cover relevant internal features of the two, brain and spinal cord also. You can see in this figure, in this photograph, a person riding a bicycle, and he is actually riding it on a hillside, so he has to climb uphill. And anybody who has ridden a bicycle on a mountainous road will know how much effort it takes to go uphill. So he is able to do this function because his muscles are coordinated by the nervous system. So and when the nervous system is degenerated or defective, so then we have this condition, and most of you are familiar with this condition. This is called Parkinson's disease. So in this disease, you can the patient is not able to do the motor functions properly, though his all other functions are fine. Only his motor activity has been diminished, and he is at some point of time he will not able to also drink water or eat food by himself alone. So all this and this both are due to the nervous system. And when you look at the nervous system, you need to first understand that how does the nervous system develop. Now this is a photograph or figure of a trilaminar embryo. So this is so the entire brain develops from a structure called neural tube, and I am going to tell you how the neural tube will be formed. And this figure is from day 7 of development during intrauterine life. So, this is the ectoderm, this is the mesoderm, and this is the endoderm. All of you know that the neural tube develops from the ectoderm. The nervous system develops from the ectoderm. And you can see there is a groove over here. This is called the neural groove. This will get thickened and ultimately it will fold upon itself to form the neural tube. The neural tube is actually a hollow tube. So let me show you further figures on this. So this is the ectoderm I was talking to you about, and it has become thickened over here to form the neural plate, and it has become the groove now, neural groove, and the cells which are at the top of this groove over here at the edges, these are known as the neural crest cells. Neural crest cells, and afterward you can see that this groove has converted into a rod shaped structure which is known as the neural tube, and these cells which were present on the margins of this groove, these have aggregated together to form the neural crest. Now these neural crest cells have large number of derivatives. The neural crest cells will form the melanocytes, which are the pigment cells of the pigment cells of our skin. This will form the adrenal medulla. This will form the dorsal root ganglia in the spinal nerve, and this will also give rise to certain parts of the heart and to the skeleton of the head and face, and it will also have a role in the development of certain a part of the thyroid gland also. So you can see that the neural tube, which is going to form the brain and spinal cord. Hello, good afternoon. Yes, yes. Yes. हाँ, मैं अभी लेक्चर ले रहा हूँ नर्सिंग स्टूडेंट्स के लिए। तो आप वो मेरे ऑफिस में छोड़ दीजिए, या आप थोड़ी मैं 4 बजे फ्री हो जाऊंगा तब आप आ जाइएगा। ठीक है, ओके। So you can see over here that the neural tube, which is going to form the central nervous system, and this the neural crest, which is going to form the peripheral nervous, the ganglion, a part of the peripheral nervous system, are developing together over here. So so you can see in this figure again that this was the neural groove which is now closing by itself to form the neural tube. So ultimately the whole thing will be closed, and it will be converted into a hollow tube, which I mentioned is the neural tube. Now, this neural tube, the anterior or the cranial part of this neural tube will show some enlargements. You can see these enlargements over here. It is not seen over here. And these enlargements are actually the sign of development of the brain in the cranial or anterior part of the neural tube. Now, when you look at this in this region, you see there are basically three dilatations, which are also known as the brain vesicles, and these are the forebrain vesicle, the midbrain vesicle, and the hindbrain vesicle. So three brain vesicles are there: the forebrain. This is the forebrain. This is the midbrain, and this is the hindbrain. This is also known as the prosencephalon, the mesencephalon, and the rhombencephalon. And this is the last part of the neural tube, which has not shown any enlargement, and this will form the spinal cord. Now, when you further look at the region of the prosencephalon, you will see that there is a very large outpouching and this outpouching is going to form the cerebral hemispheres, the largest part of the human brain. You can see these are the cerebral hemispheres in green, and you can see over here that this is going to form the cerebral hemispheres. This is known as the telencephalon, and a succeeding part of this prosencephalon is the diencephalon, and this diencephalon is going to form the thalamus, the hypothalamus, and the epithalamus. So if you just understand this, so there will be thalamus forming on each side, and just above this will be the epithalamus, and just below this will be the hypothalamus. I am sure most of you have read about hypothalamus in your 12th class, where you have seen its endocrine function. You have seen that it controls the autonomic nervous system, and you will see that it also controls several other functions like the circadian rhythm, the cycle of waking and going to sleep, the cycle of productive uh, hours during the waking period, and it is also concerned with the maintenance of the normal body temperature. The hypothalamus is also concerned with the amount of food that we ingest, the that we eat during the day. So, and people who have problems in the hypothalamus, they tend to have more and more food, so they become very obese. So the hypothalamus is a very key element, and I have just explained to you, it is a part of the diencephalon or diencephalon. And apart from that, it is also there is also the thalamus, and there is also the epithalamus. Remember, the epithalamus has a very important component called the pineal gland. Pineal gland, because it resembles the pine tree, and this pineal gland releases a hormone called melatonin, and it is also responsible for the sleep awake cycle. So, this is the diencephalon, and you can see that from the diencephalon, the second cranial nerve is coming out. That is the optic nerve, and from the telencephalon, the first cranial nerve is coming out. That is the olfactory, and then we have the mesencephalon, from where the midbrain has formed. Here is the midbrain. So this is the telencephalon, this is the diencephalon over here, and then this is the midbrain part over here, and then this is the rhombencephalon. The rhombencephalon is further divided into two parts. One is the हाँ हाँ कभी क्या है हाँ बस थैंक यू So I was telling you that this rhombencephalon has two parts. The part which is red in color is the myelencephalon, and the part which is yellow in color is the metencephalon. Now, the myelencephalon gives rise to the medulla oblongata, while the metencephalon over here, it gives rise to the pons and the cerebellum. So you can see this yellow color, it is coded over here, and you can see it has given rise to the pons, and it has given rise to the cerebellum. This is the metencephalon, and this is the myelencephalon, and it has given rise to the medulla oblongata. So we now know that the nervous system, I will just summarize what I have told you, that the nervous system starts developing from day 17 of intrauterine life in the form of a neural tube. The neural tube develops from the ectoderm. So the nervous system develops from the ectoderm, and the neural tube, as it is folding, it is giving rise to a collection of cells which are called the neural crest, and the neural crest in turn gives rise to several structures like the adrenal medulla, like the melanocytes, like the ultimobranchial body, like of the thyroid, and like the dorsal root ganglion, like the autonomic ganglion, like the autonomic neurons in the gut. So there will be number of structures which are derived from the neural crest, and you all must know these structures which are arising from the neural crest. And then we come back. This is the neural tube which is forming, and you will see that the cranial part of neural tube had some dilatations: prosencephalon, mesencephalon, mesencephalon, and rhombencephalon. And the prosencephalon is further divided into telencephalon and diencephalon. And the mesencephalon is like that only, doesn't form secondary brain vesicles. So it is an important question in the exam, that which brain vesicle does not form secondary brain vesicle. So it does not form a secondary brain vesicle, and this also forms secondary brain vesicles, that is the metencephalon and the myelencephalon, and these are giving rise to the adult brain. So here we have the adult brain with the different parts. So let us look at the external features. This is a picture of an actual human brain. It's not a model of an actual human brain, and you can see over here that this is the cerebrum or cerebral hemisphere. One cerebral hemispheres. There are two cerebral hemispheres. This is on the left side, and you can see that on the surface there are sulci and gyri. So, gyri or singular is gyrus, and this is sulcus, plural is sulci. So because the growth of this cerebral cortex was much more than the underlying part of the brain, that was the white matter, so it became folded, and in a very complicated manner, but in a very uniform manner also. So why I say uniform? Because some of the sulci and most of the sulci and gyri are roughly placed in the same position in all of us. In all of us, let us look at this sulcus. This sulcus we call as the central sulcus, and this central sulcus actually is dividing the cerebral hemisphere into a frontal lobe in front and a parietal lobe behind. The central sulcus meets over here to some extent, not completely, the lateral sulcus. This is the lateral sulcus, and this lateral sulcus is actually separating the frontal lobe from the temporal lobe. And at the back, there is a parieto-occipital sulcus which separates the occipital lobe from the remaining lobes. So this is the cerebral hemispheres having the gyri, that is the elevations, and the sulci, which some I have named, like the central sulcus, the lateral sulcus, and the parieto-occipital sulcus. And there are the gyri are also named. You can see that many of the gyri are named, but you can just remember the gyri which is present in front and behind the central sulcus. The gyri which is present in front of the central sulcus is the precentral gyrus, and the gyrus which is behind is the postcentral gyrus. So the precentral gyrus is the where the the muscles of our body are represented. So if we want to contract any specific group of muscles in our body, then appropriate part of the this motor area is activated, and then it will send an impulse through the tract down to the finally down to the spinal cord, and then or the cranial nerve to the muscles, which will then contract to bring about the action. So we call this precentral gyrus as the primary motor area. Behind the central sulcus, this is the postcentral gyrus, and just the opposite of the of this area, this is the primary sensory area of the brain. Primary sensory area means the whole body is represented over here in an upside down manner. So the head will be somewhere around here, and then the upper limbs, and then the trunk, and then the lower limbs. Here also it will be the same. The head motor area, the head, then the upper limbs, trunk, lower limbs, trunk and lower limbs. So this is called a these are called homunculus. Homunculus means little human. So this is the sensory homunculus, and this is the motor homunculus. Now let us think that as I have been using a needle, I get a pinprick with the needle. Now, as soon as I get a pinprick here in my finger, the pain travels along my hand and along the nerves to the brain. And there is a specific part in this post-central gyrus which is reserved for these fingers. So, whenever a specific part of the body is injured, or a specific part of the body is touched, or there is some stimulus, then that is going to go centrally along the sensory nerves, and it will activate a very specific area of the cerebral cortex, primary sensory area. So we know that this area has been damaged, or this area has been touched. So this is the primary motor area in front and primary sensory area behind. Similarly, at the back of the brain, this is the primary visual area at the back of the brain. So, whenever we see accident victims, we are very much careful to evaluate whether there is some injury at the back of the skull, because at the back of just inside the back of the skull is this occipital lobe, which is concerned with our central vision. So if this is damaged, then the person will become blind. Similarly, if these are damaged, then the person will suffer from paralysis, or if this is damaged, the person will suffer from some kind of anesthesia, as a result of which the person will not able to feel pain, touch, temperature, proprioception, and so many other things in the sensory area. So the point I am trying to tell you is that our brain has got specific areas which are doing specific functions. Similarly, if you look at the lateral sulcus, then this is the lateral sulcus, and just in this gyrus, this is called the superior temporal gyrus, we have the auditory area. So whatever you are listening, or the points that you are listening at this moment, the sound is actually going here into this auditory area, where the sound is then understood by means of neural pathways. So there is a primary auditory area over here, and of course you can understand that this is the sound which will also be important for understanding music. Finally, one last point. You can see over here that if this is the lateral sulcus, there is a sulcus going anterior, and there is a sulcus going just above. This is the anterior part, and this is the ascending part of the lateral, and this is the posterior ramus. We call it anterior ramus, ascending ramus, and posterior ramus of this sulcus, which is the lateral sulcus. And there is an area over here, the area 44 and 45, which is concerned with the speech area. And remember, this speech area is only on the left hemisphere. So whenever if this area is damaged due to some accident, then the person will lose the power of speech. He will become dumb. He or she will become dumb. So when injuries occur to the brain, we must be careful that the Broca's, BROCA, the Broca speech area is unharmed and intact. So this is called the motor speech area. If you go posteriorly along the superior temporal gyrus, then somewhere around here, there is an area called the Wernicke's area. And this is the sensory speech area. So whatever I am telling you, you are understanding through this area, which is the sensory speech area, Wernicke's area. So both of these are interconnected, and both of these are only present on the left side. So damage to the left hemisphere is more dangerous than damage to the right hemisphere. In this regard, I must also tell you that we have two hemispheres, left and right. the left hemisphere is concerned with the right half of the body, and vice versa, the right hemisphere is concerned with the left half of the body. So, if something happens to the right hemisphere, the left half of the body will be paralyzed and no sensations will come. Similarly, will be appreciated. Similarly, if the left hemisphere is damaged, then the right half of the body will be either paralyzed and no sensations can be felt from the right half of the body. So, each hemisphere controls the opposite side of the body. So, this is the cerebral hemisphere, and just below it, you can see this part of the brain. This is the cerebellum. Cerebellum. Cerebellum, the literally the word means little brain. So this is the cerebellum, and just in front of the cerebellum, you can see this part of the brain. This is the brain stem, and all these parts I am going to take up shortly. So when you remove one cerebral hemisphere from another, so the hemispheres are like this. You cut them through the middle, and you look at the inside. Then you see this. You see that this is the medial surface of one hemisphere, and you can see that there is a prominent bundle of fibers over here, which is called the corpus callosum. And these fibers help in communicating between the two hemispheres. So this is white matter. This corpus callosum is white matter, and it is made up of a type of white matter which is called the commissural fibers. There are two other type of fibers. If the fibers are going within the same hemisphere from one end, from one part to another, we call it association fibers. And if the fibers are coming out from the cerebral hemisphere, we call them, or going out of the cerebral hemisphere or going to different regions, then we call them projection fibers. So students, I would like to tell you that this is the, if I tell you that what is the white matter composed of, then the white matter will be composed of these three sets of fibers: the projection fibers, the commissural fibers, and the association fibers. And this corpus callosum is the largest bundle concerned with the commissural function. That is, each hemisphere informs the other hemisphere as to the exact function that is going on. And there are, you won't believe, there are about 300 million fibers in this corpus callosum. Do you know what is the number of neurons in the central nervous system? Yes, it is about 85 billion. It is about 85 billion neurons, and each neuron makes about 1000 connections with other neurons. So we can say that each neuron has about 1000 friends with which it sends and receives information. So you can imagine uh, the huge number of connections made by these 85 billion neurons, and the connections which they make is more than the number of stars in our galaxy, that the Milky Way. So you can understand the complexity of the brain and the immense number of neurons that are present over here. So now we are looking at the inferior aspect of the brain, and when you look at the inferior aspect, you can readily see this is the cerebellum on each side, and this is the brain stem. This narrow part of the brain is the medulla oblongata. This broader part over here is the pons, and still above this is the midbrain. Still in front of it, you can see this is a part of the hypothalamus, and this is the second nerve, and this is the first nerve. The first nerve, incidentally, does not go through the diencephalon. It goes directly to the cerebral hemisphere. Now, this has an important clinical and applied feature. It means that smell, the first cranial nerve is the mediates the sense of smell, and this smell is actually going directly to the cerebral hemisphere without going through the brain stem or without going through the thalamus. So, and you can also see from this brain stem the various cranial nerves which are coming out. You can see them from the midbrain, the third nerve coming out. This was the second nerve, the optic. This is the third nerve. And you can see the trigeminal. This is the trigeminal coming out from the pons. And you can see the fifth nerve over here. The trigeminal is the fifth, and then there is the sixth nerve over here, the abducens. Seventh and eighth is coming over here, and 12th is coming over here, and 9th, 10th, 11th are coming at the back. Now, when you look at the medulla, then this elevation which you see on the medulla, these are known as pyramids. And just by the side of the pyramids, this marble like elevation is called the olive. When you just dissect out the pyramid, you see there is a bundle of fibers over here, which is known as the corticospinal fibers. On each side, there are about 1 million such corticospinal fibers, which are controlling the motor functions of the opposite half of the body. And here, just deep to this, just on the back side of this pyramid is the olive, which contains the inferior olivary nucleus, and this inferior olivary nucleus has connections with the cerebellum. So whenever we are learning some new motor activity, then this inferior olivary nucleus becomes active, and it keeps on sending information to the cerebellum so that we are able to learn that motor activity in a quicker and a fast and efficient manner. Suppose somebody is learning to drive the bicycle, or somebody is learning to drive the car or something like that. So whenever we are doing some motor activity with the foot, pressing the accelerator, brake, etc., then this inferior olive helps us in learning the new motor functions. So these are parts of the medulla. The word pons actually means a bridge, and you can see that the pons is starting from one cerebellar hemisphere and going to the other cerebellar hemisphere. So you can see so many cranial nerves over here also. So I'll come back to this topic later. And you can see the in color. Now this is a diagram. You can see the various cranial nerves. Students, you know that there are 12 pairs, not 12. 12 pairs of cranial nerves, and this is the midbrain, this is the pons, and this is the medulla. You can see that from the midbrain, the third cranial nerve is coming out, and from the back of the midbrain, the fourth cranial nerve comes out. From the pons, the fifth nerve is coming out. This is the motor and sensory component of the fifth nerve, that is the trigeminal nerve. And then at the junction of the pons and medulla, anteriorly, this is the sixth nerve which is coming out, and at the side, this is the seventh and the eighth nerve. The seventh nerve has several functions, like taking the taste sensation from anterior two-thirds of the tongue, but its perhaps most important function is that it controls the facial muscles, the muscles of the face, of facial expression on one side. So each nerve controls on one side. So there is a facial nerve coming out on one side and another facial nerve coming out. Then this is the eighth cranial nerve, which again has the cochlear part and the vestibular part. Now you can see the olive, and in front of the olive, this is the 12th cranial nerve, and at the back of the olive, you can see the 9th, 10th, and the cranial accessory nerve, which is coming out. So now you know that there are 12 pairs of cranial nerves which are coming out from different parts of the brain. The first and the second cranial nerve are coming from the forebrain region, and the third and the fourth are coming from the midbrain region, and the remaining are coming from the rhombencephalon in a very specific manner. So these cranial nerves will supply the head, face, and neck, and they are doing the same functions which the spinal nerves will do for the remaining part of the body. So trigeminal, you know, the motor part of trigeminal will control the muscles of mastication. So whenever we are eating food, we are actually using the trigeminal nerve to control the muscles of mastication. Or when we are speaking also, then also the jaw is moving, and that is also controlled by the trigeminal nerve. And like this, so you please read about the different cranial nerves and their attachment to the brain stem, and uh, their name only tells you the major function. So the longest cranial nerve in the body is the vagus. See here, this is the vagus. The thickest cranial nerve in the human body is the trigeminal, and the thinnest cranial nerve in the human body is likely the trochlear nerve. But there is close competition between the trochlear and the abducens nerve. This is looking from the side of the brainstem, and this is looking at the back. Now, when we look at the back of the brainstem, we see few features, and I will start from the lower part. Over here, you can this is the medulla, and you can see some elevations over here. You can see some elevations over here. This is you need not know the elevations over here, but you need to know these tubercles. There is a tubercle over here called gracile tubercle, and there is a tubercle over here called cuneate tubercle. This is the floor of the fourth ventricle, and I am coming to the ventricles in a moment. These are the peduncles. These are the peduncles. These there are three peduncles here, cerebellar peduncles: the superior, the middle, and the inferior. So three peduncles going to the cerebellum. The superior is coming from the midbrain, the inferior is coming from the medulla, and the middle is coming from the pons. So they are giving information and carrying information from the cerebellum to the remaining part of the brain. Now, when we look at the posterior aspect of the midbrain, we see there are four elevations over here. These upper pair of elevation are the superior colliculus, and the lower pair of elevation is the inferior colliculus. The superior pair of elevation, and together these four elevations are called the corpora quadrigemina. I am sure you have read it before, but what you need to know now is that this pair of superior colliculus is concerned with visual functions and visual reflexes and the movement of the eyeballs. The inferior colliculus is a pathway in the is a station in the auditory pathway. So fibers which are carrying the sound stimulus or the stimuli arising from hearing, that is passing through the inferior colliculus. So these are the functions of the superior and the inferior colliculus. They are present at the present at the back of the midbrain, and so I go ahead and I show you how does a cross section of the brain looks like. All over the brain, you will hear that terms like gray matter and white matter. If you look at the surface, if you look at the surface of the brain, you will see, follow the pointer here, you will see that it is dark in color, and it is going like a layer, like a covering across the whole cerebral hemisphere, this dark layer. And just if you look within it, you will see the lighter whitish layer, which is having the finger like projection within this gray layer. This gray layer is the gray matter of the cerebral cortex. It is having the cell bodies of the neurons, and this is the white matter which is having the nerve fibers, and it will also have a specific type of glia called oligodendrocytes, and you know about the cellular components of the nervous system. There are neurons, and there is glia. The glia can again be of three types: astrocytes, oligodendrocytes, and microglia. The white matter is particularly rich in oligodendrocytes because many of these fibers are myelinated fibers, but it also has the astrocytes and the microglia also within it. So what we are seeing over here is when we take a section of the cerebral hemisphere, we see on the surface a covering of gray matter, and there are neurons in this gray matter, and these neurons are arranged in specific layers. We can distinguish six such layers within the cerebral cortex: layer 1 superficially to layer 6 deep. And it starts from the molecular layer, and then external granular layer, external pyramidal layer, like that. There are six layers in this gray matter, and deep to it is the white matter, and this white matter is mainly made up of nerve fibers. I have already told you about the three basic type of nerve fibers, and within this white matter again we see some gray matter. This gray matter is nothing but the basal ganglia. This is a part of the brain which is called the lentiform nucleus. This is a part of the brain which is called the lentiform nucleus. This is a part of the brain, a part of the basal ganglia, which is called the caudate nucleus. The same thing you see here also. You can see over here, this is the lentiform nucleus, and this lentiform nucleus in turn has two parts, a putamen and a globus pallidus, and this is a part which is called the caudate nucleus. So caudate nucleus plus putamen is known as striatum, and lentiform nucleus, globus pallidus, and this is composing the entire basal ganglia, along with few other parts, and along with the substantia nigra in the midbrain area. When you make a section of the midbrain, you see you see an area in the midbrain which is having dark color due to the presence of melanin pigment within the neurons, and because of that this part of the midbrain is known as substantia nigra. And whenever the substantia nigra neurons are damaged, it gives rise to the condition of Parkinson's disease, and the function of the substantia nigra is to release dopamine into this putamen and into this caudate nucleus. So you know now that the cerebral hemispheres are made up of an outer layer of gray matter, inner layer of white matter, and within the white matter again there are some groups of gray matter which is known as the basal ganglia. Now another point which I will like to bring to your attention is that you see there are cavities within the cerebral hemisphere, and you can see this cavity much better over here. These cavities are known as ventricles. Ventricles means little cavities. So these are the central empty areas within the brain which are known as the ventricles. And there are in each hemisphere there are lateral ventricles. These are the lateral ventricles. Then there is a third ventricle, there is a fourth ventricle. So CSF, within the ventricles, CSF is being produced. Cerebrospinal fluid is being produced. And what exactly produces the cerebrospinal fluid? You can see these grape-like collections of structures within the lateral ventricle. You don't see it everywhere, you are seeing it here. If you take the human brain and just put this under a magnifying glass, you will see it looks like a bunch of grapes. Actually, this has a very specific name. These are called the choroid plexus, and I will come back to the choroid plexus in some time. The CSF is being released from the choroid plexus. So the brain is a cavity because it develops from a neural tube which had a cavity in the beginning, and that cavity has persisted even in the adult, and it has formed the ventricles within the brain. So basically the brain is hollow in nature and it is filled up with a fluid called CSF. So these are from the surface of the brain you can see the ventricles inside, and this is a better figure. You can see from the lateral side, this is the lateral ventricle. Then the lateral ventricles drains through the interventricular foramen of Monro into the third ventricles. The third ventricle drains through the cerebral aqueduct of midbrain into the fourth ventricle, and the lateral ventricles are very big. It is like this in a U-shaped manner, alphabet U-shaped manner with a tail. So this is the anterior horn, this is the body, this is the posterior horn, and this is the inferior horn of the lateral ventricles. So this entire thing is the lateral ventricle, anterior horn, posterior horn, inferior horn, body, and this is lateral ventricles. There are two ventricles, one on each side, and these two ventricles are draining into a third ventricle, and the third ventricle is in turn draining into a fourth ventricle through a very narrow channel within the midbrain called the cerebral aqueduct. This is the cerebral aqueduct. So you can see over here that this is the interventricular foramen over here, through which the CSF is coming into the third ventricle through the aqueduct into the fourth ventricle. Now what is important for you to understand is that the roof, a part of the brain forming the roof of the fourth ventricle has not developed. It never develops, and in that place a foramen is present. That foramen is called the foramen of Magendie. You can see the median aperture. This is actually a place where the brain will not develop at all. So there is a opening over there through which the CSF, which was till now within the cerebral hemispheres, within the ventricles, being produced by the choroid plexus, now is able to come out of the brain. After coming out of the brain, now it encircles the whole brain. So the brain is, you can think of the brain as floating in the ocean of CSF. So the CSF which was within the brain has now come outside the brain, and we have something known as the circulation of CSF. You must be familiar with the circulation of the CSF. Now this CSF, when it comes out of the brain, it remains between two meningeal layers. You know that the brain and the spinal cord are covered by membranes, and these membranes are known as meninges, and actually they are like mothers which are protecting the brain from injury. And each brain has three mothers. The innermost one is which is closely accompanying the brain is the pia mater. Mater word means mother. So this is the pia mater, which is close friend of the brain, never leaves the brain. Then there is the arachnoid matter, and then still outside, I will show you this in a better way, and still outside is the dura mater. So what are the students, what are the meningeal layers which is covering the brain? It is the outside one is the dura mater, then comes the arachnoid matter, and then comes the pia mater. Now if you ask me that where is between which two layers is the CSF located here, then the CSF is located here between the pia mater and the arachnoid matter. We call it the subarachnoid space. The subarachnoid space. And here, when the brain has not developed, the pia mater has also not developed. So the CSF is able to travel outside. Now this is a section. This is a histological section of the choroid plexus, and you can see that there are blood vessels over here. These blood vessels are fenestrated blood vessels, means there are holes on the wall of the blood vessel, and the lining of this choroid plexus is by special type of cells called the choroidal epithelium. choroidal epithelium, which have tight junctions between them, and they actually produce the CSF. How much amount of CSF is present in the nervous system at one point of time? It varies between up to 150 ml. 150 ml. Though some textbooks now say that the amount is slightly more. So this 150 ml of CSF, which is present at one point of time, is replaced 7 times in 24 hours. So the rate of synthesis of CSF is very high, and it continuously, the CSF is being produced, and the CSF is being absorbed back into the blood through finger-like projections called arachnoid villi. So if there is some blockage in the circulation of CSF, then we get a condition called hydrocephalus. So here we have a blood CSF barrier formed by the ependymal cells which have tight junctions between them, and which have microvilli on their free surface, and which are actively producing. The E stands for ependyma, the C stands for capillaries, and you must know about this blood CSF barrier. Now, at some places, the pia and the arachnoid move very much apart. Normally, they are close. There is very little space where the CSF is there. But sometimes they move apart, creating a larger space. And where you have a larger space, we call them cisterns. So the cistern, the word cistern can be defined as large subarachnoid spaces containing accumulations of the cerebrospinal fluid. So let us look over here. The largest cistern in the brain is here, just below the cerebellum. It is called the cerebellomedullary cistern. This is the medulla. This is the cerebellum. It is present at the angle. So it is called the cerebellomedullary cistern. There is another cistern which is present over here, and this is called the cistern of the great cerebral vein. There is an interpeduncular cistern over here below the brain, and there is a pontine cistern over here in front of the pons. There are ambient cistern also, and so many other types of other cisterns also. But remember, the largest cistern in the human brain is this one, the cerebellomedullary cistern. In children, sometimes a needle is poked over here to take out a sample of CSF, but it is done very rarely and very, very occasionally. Now, this is the place where there is the opening, where the brain has not grown. This is the cerebellum, this is the medulla, and this is the foramen of Magendie over here. On the sides of this, there are another two foramen which are called the foramen of Laschka. So it is two foramen of Laschka and one foramen of Magendie are able to drain the CSF from within the brain to outside. Think of a situation where there is infection in the subarachnoid space, like during meningitis or encephalitis, where pus has formed. So what may happen is that the pus may close these openings, and when these openings are closed, the CSF is not able to drain out. And remember, the CSF has a very fast rate of formation. On an average, the entire CSF is replaced almost 7 times. About say 700 ml of CSF is being formed every 24 hours. So if there is a block, then that is a serious event, and we call it the internal hydrocephalus, because the block from the internal aspect of the brain to the outer aspect of the brain has been stopped. The communication has been stopped. So you must understand what is hydrocephalus, and you must also understand the circulation of the CSF. Now here I will show you the different layers, coverings of the brain. So if you look over here, then this is the brain. Closest to the brain is the pia mater. Then this is the arachnoid matter, and then still outside is the dura mater. When we look at the dura mater covering the brain, we see that the dura mater has two layers. One is the endosteal layer, which is lining the skull bones. This is the endosteal layer, and this is the meningeal layer. which is actually coming away from the endosteal layer. And wherever there is a need, these two layers become separate, enclosing large vein-like structures which are called as dural venous sinuses. So these are the sinuses through which the into which the blood vessels of the brain enter, drain, and these are also the sinuses into which the CSF drains. Now you know that the CSF is in the subarachnoid space, and from the subarachnoid space there are finger-like structures entering into these dural venous sinuses. And these finger like structures, these are called arachnoid villi. And when these arachnoid villi are clumped together, they are called arachnoid granulations. And these are the sites through which the CSF, which is present in the subarachnoid space, goes out into the blood within the dural venous sinuses. So if the dural venous sinuses are blocked, then the CSF is not able to drain, and again this will produce a condition of hydrocephalus. So remember, the dural venous sinuses are like veins elsewhere in the body, but the problem over here is that it is present within the dura mater, number one, and number two, there are no muscle layers within it. So it cannot undergo vasoconstriction or undergo vasodilatation. So this dural venous sinuses, there are many of them, but remember the most important probably among them are two. One is this superior sagittal sinus, which is present in the midline and the topmost part of the brain, and the other is present over here, which are known as the cavernous sinus. This cavernous sinus is actually draining the vein from the retina, and the cavernous sinus also communicates with veins from the face. So veins from the face are communicating with the cavernous venous sinus, which also is draining the central vein from the retina. Now, if there is some infection on the face, then that infection will be taken to this cavernous sinus where there will be thrombosis, and as a result of which the central vein of the retina will not be able to drain, and as a result of which the person may become blind. So we have to take care that there are no infections developing on the face, particularly in the area of the upper lip, septum of nose, and the sides of the cheeks. This is called the dangerous area of the face. And many youngsters, whenever they are developing some acne or something like that, they tend to squeeze the acne, and sometimes when they squeeze the acne, the bacteria from there spreads through the facial vein, which does not have valves to control its flow. It will flow backwards, go through the superior ophthalmic vein, inferior ophthalmic vein, and also through the pterygoid venous plexus to the cavernous sinus, which will have an infection, and the person will have high fever. There will be swelling of the face, there will be swelling of the upper lips. So please advise people not to squeeze the pustules or acne which develop during the adolescence period on the face, because they can create serious problem for the person, and if it is there, then it has to be treated very efficiently and very comprehensively with the antibiotics so the infection can be resolved. So you can see over here that the dura mater has blood vessels, and these are the dural blood vessels. And similarly, the dura mater has veins which will drain into the dural venous sinuses. The dural venous sinuses also receive the venous blood from the brain itself. You can see the cavernous sinus over here now. You can see the cavernous sinus over here. This is a very, very important sinus, one on each side. And in the center is the pituitary gland resting in an area of the sphenoid bone we call as sella turcica. So, and these are the arteries and these are the veins. Dural arteries and dural veins. The largest artery supplying the dura mater is the middle meningeal artery. If there is an impact or injury on the brain and there is some fracture or something, or the brain moves a lot, these arteries can get ruptured and they cause hemorrhage, which is called extradural hemorrhage. Extradural hemorrhage, outside the hemorrhage. If the hemorrhage is below the dura, outside the arachnoid, it will be subdural. If it is between the arachnoid and pia, it will be subarachnoid hemorrhage. So, the extradural hemorrhage are a direct result of injuries to these meningeal arteries, particularly the middle meningeal artery, and which will then compress on the brain and produce kind of paralysis in the patient. So these are the various dural venous sinuses, the superior sagittal sinus. There is the inferior sagittal sinus over here. You can see the cavernous sinus over here. Then you can see the sigmoid sinus, transverse sinus, and superior petrosal sinus, inferior petrosal. You must know the names of some of these venous sinuses. Why? Because the CSF is draining into it. Again, why? Because the venous blood of the brain is draining into this. So let us look at the lobes of the cerebrum. So these are the different lobes I have spoken to you: frontal, parietal, temporal, occipital. Each of them have different functions. Temporal, I told you, auditory function is there. Then the sensory speech area is partly over here. Then we remember faces. There is an area over here through which we are able to remember the face of the person. This is the frontal lobe. This is the area where we make our own judgments, our personality, etc. is determined by the frontal lobe. This is the occipital lobe where we have the visual area, and here is the parietal lobe where we have a representation of the space around us on this. So suppose we are seeing a kite flying in the sky, and we can actually pinpoint where is that kite flying, in which direction that is that kite flying, due to the representation, spatial representation within the parietal lobe. So this is the actual surface of the cerebral hemisphere. You can see the sum of the precentral gyrus, postcentral gyrus, superior temporal gyrus. Some of these gyrus I have already told you, they are named and they have some specific functions. So on the medial side also you have the same lobes, and you have some sulci and gyri here also. So remember this parahippocampal gyrus over here, the parahippocampal gyrus. This parahippocampal gyrus is concerned with the recent memory. So whatever you are listening, it is first going into this parahippocampal gyrus, and then it will be transferred to the remaining part of the brain. So if the parahippocampal gyrus is damaged in a person, then that person repeated long periods of alcoholism can lead to damage to the parahippocampal gyrus, and deficiency of some vitamins can lead to problem with the parahippocampal gyrus, like beriberi. You have heard of beriberi, and those patients are not able to remember things correctly. So, cingulate gyrus is also concerned with memory and emotions. So, there are so many different parts of the brain, and each are concerned with specific functions. Looking at the spinal cord, this is the spinal cord. The total length is about, on an average, it is about 45 centimeters, and it is having also the same coverings: the dura mater, pia mater, arachnoid matter. It also has the CSF between the arachnoid matter and the pia matter, and you can see that the spinal nerves are coming out from the spinal cord. There are two enlargements of the spinal cord. This is the cervical enlargement, this is the lumbar enlargement. So the spinal cord begins at the upper border of the first cervical vertebrae. What is the other name of the first cervical vertebrae? Yes, it is the atlas vertebrae. Where does it end? It ends at the level of the lower border of the first lumbar vertebrae. You know there are 7 cervical, 12 thoracic, 5 lumbar, 5 sacral, and 4 coccygeal vertebrae. So the spinal cord ends much higher up, where the junction between the first and the second lumbar vertebrae. So, but the nerves are there which are going to their respective vertebrae. So, there will be the fourth sacral nerve which will come out between the fourth and the fifth sacral vertebrae, and the fifth sacral nerve which will come out between the fifth vertebrae and the fifth sacral vertebrae and the first coccygeal vertebrae. So you are seeing over here the spinal nerve. So how many spinal nerves are there? There are 31 pairs of spinal nerves. And just as the spinal nerve comes out, you will see the spinal nerve has two roots: a ventral root which is containing motor fibers, a sensory root, a dorsal root which is carrying sensory fibers. And this dorsal root will have a enlargement which is called the dorsal root ganglion. This will have neurons which are called pseudounipolar neurons, and this dorsal root ganglia is derived from the neural crest cells. Now within the spinal cord you can see the butterfly shaped gray matter, and the surrounding it, this is the white matter. So you can see the spinal nerve formation, the ventral root, dorsal root, and they are going to form the spinal nerve, which will then divide into a ventral ramus and a dorsal ramus. Ventral ramus will go to the front of the body, dorsal ramus will go to the back of the body. When you are looking at the gray matter within the spinal cord, you see that on the ventral side, the gray matter is much larger as compared to the dorsal side. So this is called the ventral horn of the gray matter. This is called the dorsal horn of the gray matter. There is a central canal. This is the anterior median fissure, posterior median sulcus, and these are the various spinal nerves which are coming out. So we have various segments of the spinal cord: cervical segment, thoracic segment, lumbar segment, sacral segment, coccygeal segment. Just like we have for the vertebrae, there are 33 vertebrae and we have 31 pairs of spinal nerves. Remember well that the spinal cord ends much higher as compared to the lower end of the vertebral column. It ends at the junction between L1 and L2. I'll come in a moment why you need to remember this. Now this is a spinal nerve, and you can see this is the dorsal root, this is the ventral root, this is the spinal nerve, which has divided into a ventral ramus and a dorsal ramus. The dorsal ramus is going to the back, it will supply the skin, muscles, etc. of the back, and the ventral ramus will come anteriorly, it will supply the lateral side and the anterior side of the trunk. And these will also go to the upper limb and the lower limb. So you should be able to draw a spinal nerve showing the roots, showing the spinal cord, gray matter, white matter, showing the rami, ventral ramus, dorsal ramus, and to understand that initially this is carrying motor fibers, this is carrying sensory fibers. Then when they come into the spinal nerve, they mix up, and when they now are distributed via the ventral ramus and dorsal ramus, each of the ramus carries the both motor and sensory fibers. This is a cross section of the spinal cord. You can see the gray matter within, white matter outside, and you can see the spinal nerves, the ventral root, dorsal root. Actually, they have number of roots, so we call these rootlets, but ultimately they are carrying from the same segment of the spinal cord. So what is a segment of the spinal cord? A spinal cord segment, students, is the part of the spinal cord which gives attachment to one pair of spinal nerves. So you can see this is a segment of the spinal cord giving attachment to one pair of spinal nerve. So what is a spinal segment? I have told you. Now what is a dermatome? Dermatome is the skin supplied by a single part of the skin supplied by a single spinal nerve. So dermatome is part of the skin body surface supplied by a single. So this you can see one spinal segment is there. It is giving rise to one pair of spinal nerves. This is a quiz. Can you understand what is being shown over here? This is the dermatomal representation of the body. Each spinal nerve is supplying specific part of the body. So the region around the skin around the thumb is supplied by C6. The skin on the little finger is supplied by C8. So if there is some damage to the spinal cord, we can pick up where is the damage by testing for the sensory functions. So this quiz, the answer to this quiz are these figures is showing the dermatome. So you should know what is a dermatome and what is the significant. So functional components of the spinal cord, we know there are motor and sensory components. Motor component. I have already mentioned before different types of sensory fibers. And within the white matter, there are various tracks. Tracks are nerve fibers going up and down. Some are motor, some are sensory. So I conclude here, and this will be continued further by one of my colleagues who will talk to you about the blood supply of the nervous system. Thank you, students, for your patient listening. I hope you have understood it, and you will also read more about it. Thank you so much. Yes, please come. Please come. Please come. बंद हो गया? आपका नाम क्या है?
आशुतोष।